Hello, and a very warm welcome to everyone joining us today for our Select Science webinar entitled Perfluorinated and Polyfluorinated Chemicals in the Environment, What's the Big Deal? My name is Matt McCardle, and I'll be moderating today's presentation. I'm delighted to introduce today's speakers, Charles Nesland, Scientific Officer at Eurofins Lancaster Laboratories Environmental, and Zoe Grosser, Marketing Director at Horizon Technology. Today we're going to be discussing the current regulations surrounding perfluorinated and polyfluorinated chemicals, future-facing testing techniques and methods for these chemicals, and analysis of drinking water. Just before I hand over to Charles and Zoe, I'd like to mention a couple of things. Please feel free to ask any questions for the Q&A session at any time during the webinar by clicking on the Ask button at the top right of the auditorium. You can view the presentation in full screen by clicking on the tab at the bottom right of the window. The slides and demand link will be provided after the webinar. There will also be a poll question during the webinar, which you can complete by clicking on the poll link at the right of your screen. Without further delay, I'm delighted to hand over to Charles. I want to thank him and Zoe for presenting to us today. Please go ahead, Charles.
Thank you, Matt. Thanks for organizing this webinar and on this new and emerging topic. And the subject of our broadcast today, the current status for the analysis of, or at least my portion of a current status for the analysis of polyfluoroalkyl substances in the environment. Polyfluorinated compounds, and like with most environmental contaminants that we all deal with, or in our work as environmental chemists and analysts, these aren't things that just happened last week or last month or within the last year. These are compounds that we've been using for quite a few years, and in fact, they've been in use since the late 40s and early 50s. PFAS compounds, and the use of the acronym PFAS, polyfluorinated alkyl substances, is something that we've been using much more regularly in recent times to more accurately represent the broad chemistries and the broad numbers of compounds that we are being required to look at and analyze for on a routine basis. So polyfluorinated alkyl substances really incorporates both perfluorinated and polyfluorinated compounds. Perfluorinated meaning all of the carbons in that carbon chain and backbone are completely fluorinated, bonded to fluorine, and polyfluorinated in that there are a few locations, a carbon or two that has a carbon-hydrogen bond or some other chemical moiety that may render it relatively susceptible to some kind of environmental action. So PFAS compounds have been used in a lot of different applications. Many of the ones that I think we're typically familiar with are their use as additives in fluoropolymer production and surfactants for a lot of consumer applications. Stain-resistant coatings, and the one I'm most familiar with, of course, from when I was a kid, was Scotchgard applications to furniture. My mom, being one of 5 kids, 4 of them boys, my mom was extremely appreciative of a material like Scotchgard that could save her carpeting, her new carpeting and furniture from the wrath of her 4 boys. Coatings for fast food wrappers and boxes. This is the one that, when speaking with folks, when you start to think about it, it sort of makes you a little uncomfortable and wriggle in your chair. So when you think about a pizza box that may have had a light coating of a PFAS material, or a wrapper, the light coating of a PFAS material to reduce or eliminate the grease or oily material leaking out from that container. And our breathable, our outdoor products. I mean, who doesn't? I consider myself an outdoors person. Doesn't like our who doesn't like the breathable, waterproof, lightweight material that we have available to us nowadays? And it's all these polyfluorinated type compounds that make that possible. We also see these things in insecticides, lubricants, used as a mist suppression agent in chromium plating operations. And we've also seen them used quite heavily in another product, AFFF, aqueous film forming foams. So this is a product developed by 3M and the Navy in the 60s. Low surface tension, good positive spreading coefficient, was perfect for use in extinguishing fires with light petroleum products. Complex proprietary mix that's been used in pretty decent volumes for a lot of years, and you see the different applications there: military, airports, fire training installations, and we've also seen it used to protect lives from accidents in oil and gas industry and chemical manufacturing. So polyfluorinated compounds, just a little bit more of a follow-up on the applications AFFF. 3M was pretty much a sole source supplier of that from the mid-60s to 1976, and then you saw manufacturers, producers like Ansell National Foam come into the mix in 76, and you also will see names like Angus, ChemGuard, Fire Service Plus. But again, the reality is, for these materials, and as they were used, they were typically complex mixes of many and different and varied polyfluorinated compounds. And so because of that, there are a lot of different chemistries that we're looking at with regard to PFAS compounds. So we have to consider in our analysis perfluorinated carboxylic acids, perfluorinated sulfonic acids, sulfonates, then polyfluorinated telomer sulfonates. There are sulfonamides, fluorotelomer alcohols, there are fluorotelomer acrylates, phosphates, diphosphates. There are a number of different chemistries, some of which, as you can see, I haven't even listed here, that really are covered by this wide swath of description called polyfluorinated or PFAS compounds. And so here I've got a listing of a somewhat representative listing. Not that you would see this with every laboratory doing analysis. Some doing less, some doing more than this. But this is sort of a representative listing. Our current offering actually has another 5 compounds that we could put onto this list. But I give this to show you typically what might be able to be done by the one analytical technique that many of us are using, which is, and we'll talk about 537 or some adaptations to that methodology in a few moments. But here are the kinds of compounds. We see a lot of perfluorinated carboxylic acids, sulfonamides, sulfonates, many of the chemistries that I listed in the previous slide. But the compounds that are in red there are the ones that are specifically listed within EPA method 537, and we're going to talk about that, as I said, in a few moments here. And the rest are ones that actually, for many environmental projects, are being looked at or considered and actively analyzed on a regular basis. But none of those are listed as a part of this 537 methodology. So there are some limitations with the formal application of 537, and we need to be able to cover these additional chemistries, these broader chemistries. You need to be able to make some adjustments to your methodology to accommodate that. But out of these chemistries, there are 2 that I think we're all have been active in PFAS analysis that we're all very familiar with, and that is PFOA, the perfluorooctanoic acid, and PFOS. PFOA typically associated with DuPont and manufacture fluoropolymers there, although I believe a lot of the early products, DuPont was actually purchasing PFOA from 3M for use in their products until they designed and came out with a different manufacturing process. And then PFOS, principal component in Scotchgard, and the AFFF that was manufactured exclusively by 3M up until, like we had in the previous slide, 1976. But 3M has discontinued that since 2002. And I just, next couple of slides, just being a true chemistry nerd here to point out a couple of things. And number one being, we've got this carbon fluorine tail of the structure, and that's relatively common in most of the PFAS compounds that we're dealing with. But then and that's typically a hydrophobic, although oleophilic tail within the organic structure. And then we've got what I would consider the head, that carbon oxygen carboxylate group, that is a hydrophilic portion of the molecule and has some very specific implications to its behavior in the environment. And the other thing to point out on this slide is, if we look at that molecular weight, 414 AMU, if we go to most of our, outside of, say, PCB congener type work, or maybe dioxins and furans, if we go to most of our other environmental monitoring that we get involved with, 8270, 8260, chlorinated pesticides, what have you, much of that work, the molecular weight range of compounds that we're dealing with gets up to somewhere around maybe 300, one of those multi-ring PAHs that you get under an 8270 analysis. But here, PFOA, we're at a molecular weight of 414. PFOS, the structure I show here, we're at a molecular weight of 500. And in fact, some of those sulfonamides that were in the list that I had previously, we get to molecular weights that are can get upwards of 700, 800 AMU. So in environmental monitoring and analysis, this is a relatively unique proposition for us and requires some unique applications and some unique analytical methodology for addressing these compounds. And to put it in perspective for us, to give, especially as it relates to these two compounds, PFOA and PFOS, we do have some advisory or loosely regulatory limits. You see, EPA, these compounds don't have an MCL, even though they were covered under, and we're going to talk about the UCMR3 in a second here. They were covered and were part of the analyte suite under the Unregulated Contaminant Monitoring Rule number 3, which ran from about 2013 to 2015. But we don't have enforceable MCLs, or like we do for many other organic contaminants and constituents. But EPA in May of 2016 came out with health advisory levels for PFOA and PFOS, and that would be 70 for one, 70 for the other, or 70 combined. So any combination of PFOA and PFOS that's at or above 70 would exceed the health advisory level. And while that gives somewhat of a line in the sand, that is that's not a hard line. It's not a specific regulated level. You see, I've got some examples of limits listed for a few of the other states. A couple updates. Minnesota, I think, recently has updated theirs. I need to update this slide with what their new proposed limits are. New Jersey just yesterday or the day before, I think it was yesterday, actually signed into law a legislated value for PFOA of 14 nanograms per liter, and for PFNA of, I believe it's 13 nanograms per liter. And those now will become, and I believe this is the first instance of this in the U.S., those will now become regulated drinking water concentrations or levels in New Jersey for the state of New Jersey. So they will become New Jersey drinking water MCLs. So that's been established for PFOA and PFNA. And you can see Vermont has a standard at 20 parts per trillion for each, and they've adopted the same kind of approach that EPA has. It's just at a different concentration level. It's 20 for the one, 20 for the other, or 20 combined. And then just for perspective, I put some action levels for EPA Region 4 in soil, and you see those are in the ppm range. But there's really only one sanctioned method for doing this analysis, and that is EPA Method 537 version 1.1. It's a drinking water method, so it's relatively prescriptive. It's got a prescriptive list of compounds, prescriptive way in which you handle and process the sample. There is some method flexibility allowed, but as with most drinking water methods, because in most cases those drinking water methods are developed and intended to be used for regulated, water supplies and regulated compounds in water supplies, they're relatively prescriptive. But one of these really positive things that came out of this 537 was the use of LCMS-MS, which is a powerful analytical technique. And prior to its implementation in 2009, the use of LCMS-MS in the environmental industry was relatively limited to just a few of we laboratories that were either using it for some PFAS kind of work, some early PFAS work, or some other method development kinds of applications. But as I alluded to when I had the slide up there with the list of compounds, 537 has some limitations in the range of compounds that can be accommodated. It uses, and part of that is because in that it's using a styrene divinylbenzene solid phase extraction cartridge, adequate for it's a fit for purpose kind of situation, adequate for the list of compounds in the method. But when you then start to add in or need to broaden the scope of compounds, like we see in many environmental investigations now, as these events are evolving with regard to PFAS contamination in the environment, we need to look at these additional compounds and things like the C4, the butanoic acid, the pentanoic acid. The method as it's written struggles with those compounds, struggles to retain those compounds. They break through on that styrene divinylbenzene extraction cartridge and under the set of conditions for doing the extraction. Also, I would argue, and there's some active discussion within the environmental community right now, I think, but EPA is pretty much designed. EPA 537 is pretty much designed like 8270 in that it uses three surrogates. So you spike in three surrogates that are similar, but not exactly the same as all the compounds that you're analyzing for, and you use those three surrogates to recoveries of those three surrogates to describe the performance of the other compounds. And then three internal standards, so you're using an internal standard calibration technique. 537 was used extensively for the Unregulated Contaminant Monitoring Rule, UCMR 3, and here I've got listed the contaminants for under UCMR 3, and also some of the MRLs, minimum reporting limits that were prescribed or used for the UCMR 3 surveys. And for those of you, just to say very quickly, without spending a lot of time on it, UCMR 3 survey basically covered water supplies that were in excess of served in excess of 10,000 residents. So smaller water supplies didn't get covered, and also we could argue that these levels, compared to what most of we laboratories can do, these levels are relatively high compared to what most of we laboratories are routinely reporting. And I give an example for some of the compounds here of what laboratories are routinely reporting. You can see for waters, for most of these compounds, we're at down at 2, and that's an LOQ. And in fact, I would argue 2 and a couple of the compounds that don't perform as well are a little bit higher than that, but 2 parts per trillion. And I would argue we're not really even pushing really hard with that. And then soils, you can see we're at just below a part per billion, whereas that EPA Region 4 guidance was in the ppm kind range. You'll also see a few other methods referenced. 2501, there's currently some work going on to do a method validation for the ISO method 25101, where they've increased the volume, the number of compounds to their analysis list, and have made it an isotope dilution technique. ASTM method, the 7979, is a sample that's a direct injection analysis, so it has some attraction from that standpoint. But it uses, in my opinion, one of the conflicts with what we think is the way to approach this analysis is that it uses an external standard calibration as opposed to isotope dilution. And I'm going to talk about that here in a second. So as I indicated, I think before, most methods are typically fit for stated purpose, but may fall short when you go to a wider range of compounds and matrices. And for waters in general, we're always using solid phase extraction. Soils, solvent extraction with a water and water-soluble solvent mix. And I talked a little bit about, I think I'm going to bump through this slide because I'm afraid I'm running long on time, but 7917 avoids that extraction by direct injection, has a broader list, so we're up to 31 compounds, use of 14 isotopically labeled compounds as surrogates. So that's a good approach, but they're still strictly as surrogates, and we've got reporting limits in that 10 to 25 nanogram or part per trillion range for most compounds. I think the argument that we're trying to make is that for our applications, we're tasked for, when we look at a broader range of compounds, wider range of matrices, water, wastewater, landfill, leachate, soils, sediments, tissues, high organics in samples. We've applied weak anion exchange for extraction and cleanup, and have also upped it to 24 isotopically labeled compounds that have a dual functionality. They function as a surrogate and as the internal standard used for quantification. So we've got the benefit and we've got the power of that isotope dilution, where we're auto-correcting for recoveries within sample matrices. We've also put in injection standards so that we've got some monitoring of the QA/QC for the sample extract versus the sample extraction. These injection standards then are also used to quantify the recoveries of the isotopes that we're using for the isotope dilution. So again, we get the dual functionality. We can calculate an absolute recovery, so it shows an extraction efficiency, but then at the same time, that compound is used for the quantification of its native analog. So you get an auto correction like you typically do for your isotope dilution. And this is a practice when we talk about isotope dilution. Sometimes people are not familiar with it and think isotope dilution, that sounds like this big complicated analytical chemistry thing. Well, I mean, we've been doing isotope dilution for high-res mass spec type work for dioxins and furans and some of our PCB congeners for lots of years, and those methods are 25, 30 years old, and so we've got lots of applications where it's been used quite well and quite effectively, and I think this is an appropriate place for it also. So we've got a target list of 32 natives that are covered by 24 labeled compounds, isotopically labeled, and this is typically a C13 type compound. Not all, but most are C13 labeled compounds. And so our limitation here is really what's available from standards manufacturers. If standards manufacturers, once they are able to supply additional labeled compounds that match with each of the native compounds that we're analyzing for, then it can become a full isotope dilution analytical technique. And so I know this slide's a little busy, but I just wanted to give everybody an example, a visual example of compounds or data that's reported. So we've got at the top of the first column there, we've got the names of our native compounds. So there's PFBA, butanoic acid, a pentanoic acid. There's a fluorotelomere sulfonate, 4,2 fluorotelomere sulfonate, all the way down through. And then if we go to the 2, 3, 4, 5, 6 column over, and it has IS named there. There we've got each of the isotopically labeled compounds that are used for, number one, as surrogates, and that also function as the internal standard for calculation and quantification of the native compound recoveries. So in September of 2016, EPA released a document with regard to quantification for PFOA. And I think this came about because of a situation that they were in where they were splitting samples between two laboratories, and they saw a consistent 20% difference. I'll use the term bias, but I think really just to say it different, 20% difference between the results for PFOA between these two laboratories. And as they drilled down in to try and understand why was there this consistent 20% difference, they determined that the one laboratory was using linear isomer PFOA for their calibration standards, which most of us are doing, but then identifying that peak in the sample and quantifying the linear isomer of PFOA. The other laboratory recognized that there were, in environmental samples, that there are both linear and branch chain isomers available, so they were lumping the areas of both the linear and branch chain isomers in the sample and then quantifying it with their linear calibration standard. And this resulted in approximately a result that was approximately 20% higher than the other laboratories. So EPA came out with this guidance on how to handle quantification and analysis of PFOA so that we've got some level of consistency in the results that are reported. And here, I'll just give you a graphic of what that looks like. On the right-hand side, that's actually the carbon-13 labeled analog of the hexane sulfonate, but you can see it's a single peak. And then if you look to the left, you see that similar or same single peak, that's the native hexane sulfonate, but that little hump that's right in front represents the branch chain isomers. Now, in the chromatography that we're using here, we're not looking to try and separate out all the branch chain isomers because we're not looking to delineate all those branch chain isomers, but we want to see it well enough so that we can do a quantification for those branch chain isomers. So just a comment on certification, and I think I'm getting close to the end here, Zoe, but the certification world is still in flux for many states with regard to PFAS analysis. Again, it's complicated by the fact that there's not an enforceable MCL other than New Jersey's action from, like I indicated, I think just yesterday. States are offering certification or doing so by 537, which is drinking water, but it's not really applicable to certain groundwater matrices, wastewater, surface water matrices, and certainly isn't directly applicable to soil, solid, or tissue analysis. But we, in reality, are in situations where we're having to analyze for all of those different matrices. So that's wherein our argument is the application of a modified, and I know most folks don't like hearing it this way, but a modified EPA 537, where we're doing isotope dilution, we're using a different kind of extraction cartridge that covers a wider range of chemistries, and incorporating a couple other parameters to allow us to handle this wider range of chemistries. And then the big thing that has happened that's a big bonus is that a proficiency program for PFAS just started in January of 2017. Those of us in the environmental industry, we've had this in place for 8270 and 8260, 6010, and 6020 for years, for lots of years. And so clients and we laboratories could always go to the PTs as an indication of overall performance and method performance. We didn't have that for PFAS, but we just started with a commercial offering of that in January, and so now that that is well underway, I think we'll be able to get a better handle on the proficiency of each laboratory's application of analysis for PFAS. And then to point to another element of this PFAS analysis world, the DoD released in January 2017 QSM Quality Systems Manual 5.1, and in that, while it's not a method, it's not a PFAS method, but it is a set of quality criteria that should be an umbrella or should be a framework around which or within which your PFAS analysis method can operate. So it defines things, stipulations about the calibration, things about the order and concentration of your CCVs. They give some prescriptions about what should be done with cleanup of samples, sensitivity monitoring checks, several other QC criteria. So it gives that framework against which, even though Lab A and Lab B and Lab C may have slight different variations in the method that they have set up for PFAS analysis, as long as each of those slight different variations can meet the criteria of this QSM 5.1, then they potentially are a viable certifiable under the DoD criteria method, which I think has some powerful application for us. And then my last slide here is just to point out some things to try and avoid when sampling. I think we're at a point where we're filtering through some of the old wives' tales that might have been out there with regard to sampling. I think a joke that you typically hear at a lot of conferences that we go to is, I believe it was an Air Force document from maybe 3 years ago, that when you basically read through it and all the do's and don'ts and what you can't use and personal care products and this and that, I think one came away from reading that thinking, gosh, to go sample for PFAS samples, I basically need to go out and do my sampling naked, and I can't have taken a shower or bath for several days. So it doesn't really need to go to that extreme, be quite that extreme, but there are a few things that a number of laboratories have looked at and determined that they do potentially have some residual concentrations of PFAS compounds, and that would be the chemical ice bags, some Post-it notes, waterproof field books. We say generically equipment using Teflon. But I will tell you that we've had some tests where we see some small residual background, and then we've seen others where they're squeaky clean. So it's a mixed bag. So because of that, you basically need to test and make sure that what you're using is clean. Tyvek suits, sunscreens, and insect repellents can have residual levels. Sharpies, aluminum foil, new clothing. Probably a rule of thumb, because you know most all of our clothing has some kind of stain-resistant kind of material impregnated in it. Rule of thumb is wash it multiple times before you use it in the environment, and then any clothing laundered with fabric softener. So with that, I'm going to hand it over to Zoe, and I thank everyone for your attention.
Thank you very much, Chuck. I'm going to talk a little bit about solid phase extraction, since that's a mechanism used in most of the methodology for the PFAS compounds. One of the things that folks have mentioned is that dilute and shoot is an easy way to do a lot of this work. You don't have to do any sample preparation. You just dilute, shoot it right into the mass spec. However, sample pre-concentration and cleanup has an important place, and that's what we're going to talk about. So generally, for environmental solid phase extraction, you see either discs or cartridge formats. You won't see some of the things that you see in pharmaceutical or biotechnology, where you might have 96-well plates or SPE in a cartridge tip. It's basically discs and cartridges for this type of work. So discs are excellent when you're using larger volumes of liquid or liquid with particulates. So, for example, if you're doing a wastewater sample, as Chuck had mentioned earlier, then something like a disc is going to be a very convenient format for your SPE adsorbent. Cartridges are convenient for smaller volumes and when samples are relatively free from particulates. So, for example, in 537, the drinking water method, it is a cartridge that's used and it's 250 milliliters of water. So, on the bottom of this slide, you'll see a comparison of a cartridge and a disc in terms of size, and also a little abstract for a food analysis that was done using SPE adsorption of the analytes and then an analysis. So I think that this is even going to move into food as we move forward. So it's not just going to be environmental concern with the PFAS compounds, but also things like food where people will be exposed to the compounds. Now, this slide gives a little bit of a comparison of cartridges and SPE discs in a little more detail. So here we're talking about cartridges where the flow rates must be controlled in order for the kinetic activity to take place. And that's pretty important. One of the reasons why automation is important for cartridges more than anything else is because the flow must be controlled, and that's very difficult with a vacuum manifold. Discs are great in terms of flow rates. And we're going to show on the next slide a little bit of the reason for that. The kinetics can happen very quickly because the particles in the disc tend to be smaller. So you get a lot of activity between the sample going through the disc and the disc particles, whereas cartridges, the particles may be a little larger, and you have to make sure that the effects that you want to take place have the time to do that. Now, we'll mention also, and here we have our poll question. Okay, so we're going to push out a poll for you to answer, which says, in your opinion, what would be the biggest advantage of solid phase extraction in your lab? We went over a few of the definitions, so what do you think would be the biggest advantage in your lab? Less attention required to get good results? Is it better reproducibility? Is it speed of analysis? Or lower cost? And when that poll box comes up on your screen, you can click on the answer that is most appropriate for your laboratory. So the choices are less attention required to get good results, better reproducibility, speed of analysis, or lower cost.
Zoe, if you don't mind, we can carry on and we'll come back to the poll at the end when we publish it, if that's okay.
That would be fine. I see the results here, and it's quite interesting. I think it will be interesting for everyone to see that. I think everybody has a good handle on how SPE works in their laboratory. Okay, we'll go on to the slide on the Stockholm Convention. I think that it's important to realize that this is a global issue. It's not just a U.S. issue. It's a global issue, and there's a lot of concern in Europe about these persistent chemicals. So anything that the Stockholm Convention addresses is also going to be important to everyone else and will have worldwide impact. Now, as Chuck has mentioned, this started out with a small set of compounds with the drinking water method, but people want to do more compounds, and there are some issues with that. So we've been working with a Swedish university, the Swedish University of Agricultural Science in Uppsala, and trying to help them to do a better job of looking at 8 to 10 liters of seawater at a time. They're looking at very, very low levels of PFCs in seawater and want to be able to concentrate that down and measure it and understand what the ambient concentration is. So we worked with them to develop a disc with weak ion exchange media that might be helpful, and they're working on evaluating that, and we'll continue to work with them until we get the proper media for this work. But a disc is going to be much faster for 10 liters of seawater than a cartridge would be. So this is a pretty interesting experiment that they're doing. And here are some of the results for just a few of the many compounds that they're looking at. And this compares the HLB media from Horizon with a weak ion exchange media, and they tried two different flow rates for the weak ion exchange, anion exchange. And that's important when you're doing 10 liters, because it may be possible to have breakthrough if you go too quickly with that much water. And here you can see that you do seem to get better recovery with the slower flow rate. So it's a study that we'll continue to work with them on to optimize for the wide variety of compounds that they want to look at in a very, very large amount of water. Now, one of the things that's important always is automation, especially because you get good reproducibility, especially when compared to liquid-liquid extraction or between different analysts. It's modern technology. It's more hands-off, so you don't necessarily have as much issue with contamination. And we know that the PFAS compounds are everywhere, so contamination is a problem. So as I said, we'll continue to work with this group to understand how to make this analysis more rugged and to continue to improve the performance. Now, this was a newspaper that one of our service engineers picked up in New Jersey and took a picture. 1 in 5 in New Jersey drink water laced with PFOA. This is an issue, and people are concerned about exposure to these compounds. So as Chuck mentioned, there are a number of methods out there, but really US EPA method 537 is the one that people are using most frequently, and this uses a styrene divinyl benzene cartridge, 250 milliliters of water for a suite of PFCs, and the analysis is HPLC, mass spec, mass spec. Certainly in Europe, they're working on the ISO method, so there's a lot of attention to methodology. Now, SPE cartridges are used for this method, and when cartridges are used, you have to be very careful to have consistent flow rates so that you do get the best performance. So that's one of the things that we've worked on automating with a Smart Prep cartridge extraction system. And again, it's very hands-off so that you don't have to worry so much about contamination from things as you go. The Smart Prep automates the extraction process and has precise flow control. So this is particularly important when you're looking at trace levels and you want to make sure that you don't have any contamination coming into the system. I know Chuck mentioned that having PTFE in your instrument isn't necessarily a fatal flaw. We've tried to remove all PTFE that we can from the system. So we have FEP tubing to eliminate sample contact with anything that may contaminate it. And then you can always rinse with a little bit of methanol, too, before you run a sample to be sure that you have gotten any active PFC on the surface away. So we worked with Northern Lake Services, a lab in Wisconsin, for their implementation of 537, and this is their MDL data, and you can see it's kind of tiny on the screen, but they had very, very low MDLs calculated, so certainly they're going to be able to look at the limits that we talked about for the health advisories, so that shouldn't be an issue. This next slide just shows a little bit more detail about their laboratory control samples, spiked at low levels, parts per trillion, and the recoveries they were able to get. So again, they have good method detection limits. They have good spike recoveries at low levels. Now, in terms of laboratory fortified sample matrix and the matrix duplicate, again, very good relative percent differences, well under the 30% criteria that was specified in the methodology. Now, method 537 contains 14 analytes, and what we were looking at was the first 6 analytes that Northern Lakes had implemented. They've now added analytes to their method, so they're looking at 12. But we've also worked with Eurofins, Eaton Analytical for the full suite of analytes that are in that list. So Horizon Technology has demonstrated low background with the list, even after the introduction of moderate concentration samples. Certainly carryover is one of the things that people are concerned about. I mean, you can show that you can get low levels once, but if you have any sort of carryover, it can be a problem. So there are 2 compounds that are a little sticky, and when high concentration samples were introduced, additional clean-out time was required. And that's something that people aren't thrilled about. People would prefer that the cleanup happens quickly, and therefore their cycle time stays low. And this just shows some of the results from the initial demonstration that we did with Eurofins Eaton Analytical, and it shows that the concentrations were extremely low. So what they're looking for is a 2 nanogram per liter reporting limit. So their blanks have to be below 0.6 nanograms per liter. So that provides quite a challenge. And we were able to meet that, except that there were a couple of compounds that were a little sticky. So we are doing some additional work with another laboratory in this case to maybe make some modifications to our Smart Prep system, develop a new accessory to speed sample loading, and to maintain performance and reproducibility at the same time. And that's always a challenge with cartridges because you're limited by how fast you can put your sample through the cartridge. But we think we can do a little better, and then that will help to reduce, it will improve cleanout and help to reduce the cycle time so that the sample throughput is improved. So to conclude, there has been lots of research into methods and matrices for perfluorinated and polyfluorinated compounds, and still a lot of work to be done. I mean, Chuck said that, and I have as well. There's a lot that needs to be understood about how much needs to be examined, how much water you need, what other kinds of matrices will be involved, what compounds will eventually be shown to be the most of concern. So the US EPA, the Department of Defense, and the European Union are concerned about exposure to these compounds from drinking water in particular and food, and then that will expand certainly to wastewater, which eventually becomes drinking water. So although less sample preparation speeds analysis and is useful for screening, solid phase extraction provides a cleaner, more concentrated sample for analysis. And cleaner samples help to keep the LC-MS cleaner longer. And with that, I will turn it back over to Matt for the question and answer session.
Brilliant. Thank you so much, Zoe, and thank you, Charles, for that interesting seminar. Let's go back and have a quick look at the poll results before we go on to the question and answer seminar, if that's okay, Zoe.
Great.
So the question was, in your opinion, what would be the biggest advantage of SPE in your lab? And we got 41% of people, nearly 42, said better reproducibility was a big factor for them. 32% said speed of analysis, and 13% of both lower cost and less attention required to get good results. Is that what you would expect, Zoe, Charles?
I think that is very representative. I think better reproducibility is a critical item, especially when you don't want to have to rerun a sample. So that's exactly right. So it makes sense to me.
I'm sorry, Zoe. I agree, and in fact, I'm a little bit surprised. Anyway, I think people tend to typically think speed of analysis, that we laboratories, we're always looking at how fast we can put it through, because more samples means more in revenue. But the reality is, in the long haul, it comes back to the better reproducibility, which that is the key. So if you've got consistent results, which then relates to good quality results and quality reporting, that over the long haul is actually probably the better parameter, the better throughput.
Brilliant. Thank you both. Let's move on to the last part of today's webinar, the question and answer session. We've got a few questions in here. The first one is from Mary, and it is, can you expand on some or all of the acronyms you use in the presentation? DoD, CCVs?
I'll take that one. Sorry, Mary. Guilty as charged here. DoD, Department of Defense. I don't know if you had some other specific ones, but DoD was Department of Defense. QSM was Quality Systems Manual, and that's related to the DoD Department of Defense systems on quality. PFC, Perfluorinated Compounds. I guess if you have a few others, maybe type in a question on a specific one that I didn't cover.
Thank you for that, Charles. We've got another one. Will parafilm used to temporarily cover samples kept in beakers have PFAS? I think that one's maybe for you again, Charles.
You know, that's a good question, and I don't know, because we, in our laboratory, for virtually all of our environmental testing, do not use parafilm. So we haven't really spent any time looking at it or worrying about it. Some of the other, like aluminum foil and some of the other materials that were on the one slide, we do, and so we've evaluated those. But parafilm, we haven't used, and so I don't have any experience with that. Zoe, have you folks done anything or looked at anything with parafilm?
No, we haven't. I wouldn't have the first guess on that.
Yep.
Not to worry, Zoe. I've got one I think aimed at you. So when would you choose SPE discs versus cartridges?
Well, I think that both can have the same kinds of media. However, when you have a large volume of sample, the extra surface area of the disc is a real advantage for having the sample go through the disc quickly. It's also great for capturing particulates. You don't want to pre-filter a sample and remove the particulates before you run it through the SPE disc, because then you may miss some of the analytes that are absorbed on the particulate. So it's much better to have them sit on top of the disc and then be rinsed with the solvent in the elution step. So I think that's the main difference between them. The cartridges for smaller volumes and also for samples that have no particulates, like drinking water, are acceptable.
Thank you. We've got a question from Vivek. Is there a limit or advisory level for total organic fluorine?
I'll take a stab at that, and that it's a simple, not that I'm aware of, Vivek. There are some, just sort of take a bunny trail off of that, but there are some analytical techniques to potentially address PFAS, overall PFAS contamination, that look at total fluorine, like PIGY, proton-induced gamma emission spectroscopy, and there's also another application, absorbable organic fluorine combustion ion chromatography, which are attempts to look at PFAS, maybe a screening of total PFAS contamination by getting at the fluoride content. But as far as a regulated level for total organic fluorine, I'm not aware of one.
Thank you. So I've got one from Jack here. How do you minimize contamination?
Well, I think that contamination is an issue that you can minimize by using an automated piece of equipment, because you have less hands-on, so you're not manipulating the sample as frequently and have less chance of contamination. In addition, if your instrumentation is pretty free from Teflon and other things that might contain these compounds, then you're going to keep your sample clean. And as I mentioned, if you just put a little rinse of methanol through before you run your sample, that will help as well.
Thanks, Zoe. Do you think these types of measurements will be more often required in the future?
I think that one's for you, Chuck.
Well, I think yes. I think the one thing that I indicated in my part of the presentation, where just, and I might be off by a day, but I think it was just yesterday that New Jersey Governor Christie signed into law an MCL, a regulated drinking water level for PFOA and PFNA. So they have hard targets now for drinking water in New Jersey for those two compounds, as opposed to the health advisories that EPA has, which don't really, they sort of give a line in the sand, but they don't have any teeth associated with them. But we see this out of New Jersey, and at conferences I've been to over the last couple months, we see states in the Northeast in particular setting up and establishing some guidelines for regulating these compounds and these classes of compounds. So I think the answer is yes, and I think we're just at the very beginning of this journey.
Thanks, Charles. We've got one more from David, and this is: with the Smart Prep solution, is the whole sample used and the container rinsed for quantitative transfer of potential analyte?
Yes, that is the case. I mean, the Smart Prep is a flexible piece of equipment, so you can set it up different ways, but the way it's set up right now at Northern Lake Services is exactly as you're asking about. The whole sample is used and the container is rinsed, so there should be nothing left sticking to the glass.
Brilliant, thank you. Well, I think that's all we have time for today. I'd like to thank Charles and Zoe for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I hope you found this a worthwhile session. If we didn't manage to answer your questions, we'll follow up with you personally. If you have any more questions, please feel free to email me at editor@selectscience.net. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available on our website shortly. Goodbye and thank you once again.